Singh, and welcome to this course. I guess we'll wait for 3 to 5 minutes so that students can join in, and then we can start our today's discussion. If you have any problems, any questions, feel free to ask in the chat box, or you can directly ask me also. So we'll wait for 5 minutes and then we're gonna start our today's session. Okay. We'll wait for 1 more minute and then we're gonna start our session. Okay. Hi Gayatri. We are waiting for 1 to 2 minutes so that more people can join in, and then we're gonna start our today's session. If you're comfortable in the meanwhile, you can turn on your camera, introduce yourself, basic information. Yeah. You can also type in the chat box so that I know where are you from, what course you are pursuing. Let's make it a discussion and interactive session. Thank you, Gayatri. So if you can unmute, it will be nice to have a conversation. Yeah. So you are in which year of your masters? You started your masters or you are about to complete your masters? Okay, that's great that I'm audible. So, I don't think so. You were there in the previous class. So, I'm gonna just give a very, very, very brief discussion what I have discussed in the last class. It was very primary discussion. I basically discussed about what I'm gonna cover in the course. As you might have already received the flyer, the course content are there. So, I just discussed basically in what module I'm gonna discuss what, and overall I discussed some basics about cell membrane. So what exactly is a cell membrane, right? So cell membrane, it defines the periphery of a cell, separating its content from the surrounding to the interior of a cell, right? And then this is one of the representative and the initial images of a cell, and this is the zoomed in image of a cell membrane, how exactly it looks like. We're gonna discuss in the coming sessions that what exactly are these round things, these thread-like structures, or these gray blob-like structures. Though, as most of you were from biology background and are pursuing masters and everything, you guys already know what somewhat have information about what a phospholipid is, what is a biological membrane, what is a fluid mosaic model. But still, we're gonna discuss it in kind of details so that you have more understanding of a membrane. In the last class, we also discussed about why we need to study and understand the cell membrane as a whole, uh, uh, why we should know in depth about the membrane. Overall, we have connected the dots of understanding the biology and the physical properties of a cell membrane by connecting it with the functionality of a cell. Okay, so it's not a huge it's not a like very far connection, it's a direct connection because our cell basically interacts with the external environment via the proteins which are present in the cell membrane itself, right? So cell membranes actually play a very crucial role in maintaining and keeping a cell alive, at the end of the day keeping us alive, right? So some of the functions we have discussed in the previous class regarding functions of a cell membrane. For example, cell membranes serve as a barrier and gatekeepers. This I guess most of you are aware of, and you guys will agree that if a boundary is associated with something, it will definitely act as a barrier, and it will separate out the contents which are inside the cell membrane from the contents which are outside the cell membrane, right? They are semi permeable, which means that some molecules can come across the cell membrane, which is lipidic bilayer in nature. Don't worry, we're going to discuss what lipid is, what bilayer is. Okay, so small hydrophobic molecules like gases such as oxygen, carbon dioxide can easily yeah, let me hit the laser pointer. Yeah, can easily pass through the membrane, while polar molecules and large molecules cannot easily pass through the membrane. This is the reason for these molecules we need specialized proteins which help in aiding the transportation of these substrates, right? And cell membranes basically restrict the diffusion of highly charged molecules such as ions and large molecules. You guys must be aware that why this is important, because there is a potential difference across a cell membrane which needs to be maintained for the proper functioning of a cell, right? So we're gonna discuss and we're gonna touch on all these points in the gradually throughout the session. Okay, so the passage of these molecules relies on specific transport proteins embedded in the membrane. So now we have some idea about what is the need of having a protein like these gray blobs. These are basically the proteins which are present in the lipidic bilayer of a cell membrane. So why these proteins are there? These proteins are there to assure that that small molecules, large molecules, polar molecules, they have a space and place through which they can be easily transported throughout the cell. It can either be transported outside the cell, or they can be imported from outside to the inside of a cell, depending upon the need of a cell. Okay, and then in the last class we also discussed about a very brief history of a membrane structure, because as we know that the model which is acceptable for the membrane structure is the fluid mosaic model. So it is not a thing that fluid mosaic model directly came into picture. Fluid mosaic model came into picture before by adapting to the models which were gradually spread throughout the history. The structure of a membrane, or the lipidic composition, that the membrane is actually of a lipidic nature, was came across in 1925, right? Then other models such as Pauci model, unit membrane model, and then finally the fluid mosaic model, and the recent one which is the lipid raft model. So lipid raft model is basically a fluid mosaic model, is just also describes the occurrence of microdomains, right? Microdomains, or which are called lipid rafts, because they are highly concentrated of particular type of lipid and particular type of protein. So everything is encompassing the properties of fluid mosaic model in the lipid raft model. The additive point in the lipid raft model is the occurrence of microdomains, okay, which the fluid mosaic model was not able to comment on. I guess I'll upload the session, hence I'm not discussing a lot about what I've already discussed in the previous class. So for today we're gonna start by watching certain videos which will help us in understanding some basics of membrane, and then we can build our understanding on them. Okay, so let me share
as a software
my
developer
screen with you guys.
Have you ever been curious?
Just let me know if you can see my YouTube screen and you can hear it also. Okay, I hope that you can see it.
Accuracy?
Yeah, so this video is basically will tell you about very basic structure of a cell membrane. So if you do not know some of the terminologies, don't worry about it. You can ask questions in the chat box, or you can wait till the end of a class. Maybe most of your doubts might get cleared. Okay, so I hope that you can see as well as you can hear the particular video.
Cell membranes are structures of contradictions. These oily films are hundreds of times thinner than a strand of spider silk, yet strong enough to protect the delicate contents of life. The cell's watery cytoplasm, genetic material, organelles, and all the molecules it needs to survive. How does the membrane work, and where does that strength come from? First of all, it's tempting to think of a cell membrane like the tight skin of a balloon, but it's actually something much more complex. In reality, it's constantly in flux, shifting components back and forth to help the cell take in food, remove waste, let specific molecules in and out, communicate with other cells, gather information about the environment, and repair itself. The cell membrane gets this resilience, flexibility, and functionality by combining a variety of floating components in what biologists call a fluid mosaic. The primary component of the fluid mosaic is a simple molecule called a phospholipid. A phospholipid has a polar, electrically charged head, which attracts water, and a nonpolar tail, which repels it. They pair up tail to tail in a 2-layer sheet just 5 to 10 nanometers thick that extends all around the cell. The heads point in towards the cytoplasm and out towards the watery fluid external to the cell, with the lipid tails sandwiched in between. This bilayer, which at body temperature has the consistency of vegetable oil, is studded with other types of molecules, including proteins, carbohydrates, and cholesterol. Cholesterol keeps the membrane at the right fluidity. It also helps regulate communication between cells. Sometimes cells talk to each other by releasing and capturing chemicals and proteins. The release of proteins is easy, but the capture of them is more complicated. That happens through a process called endocytosis, in which sections of the membrane engulf substances and transport them into the cell as vesicles. Once the contents have been released, the vesicles are recycled and returned to the cell membrane. The most complex components of the fluid mosaic are proteins. One of their key jobs is to make sure that the right molecules get in and out of the cell. Non-polar molecules like oxygen, carbon dioxide, and certain vitamins can cross the phospholipid bilayer easily. But polar and charged molecules can't make it through the fatty inner layer. Transmembrane proteins stretch across the bilayer to create channels that allow specific molecules through, like sodium and potassium ions. Peripheral proteins floating in the inner face of the bilayer help anchor the membrane to the cell's interior scaffolding. Other proteins in cell membranes can help fuse two different bilayers. That can work to our benefit, like when a sperm fertilizes an egg, but also harm us, as it does when a virus enters a cell. And some proteins move within the fluid mosaic, coming together to form complexes that carry out specific jobs. For instance, one complex might activate cells in our immune system, then move apart when the job is done. Cell membranes are also the site of an ongoing war between us and all the things that want to infect us. In fact, some of the most toxic substances we know of are membrane-breaching proteins made by infectious bacteria. These pore-forming toxins poke giant holes in our cell membranes, causing a cell's contents to leak out. Scientists are working on developing ways to defend against them, like using a nanosponge that saves our cells by soaking up the membrane-damaging toxins. The fluid mosaic is what makes all the functions of life possible. Without a cell membrane, there could be no cells, and without cells, there would be no bacteria, no parasites, no fungi, no animals, and no us. TED-Ed has
Yeah, so in the video, as I've told you, that was a very basic I'm sorry, there's some issue with my Teams app. Yeah. Yeah, so hi Shrish. No worries. It will be great if you can just put your basic introduction, like what you are doing, where are you from. It will be nice to have a conversation and discussion. I know most of you were not there in the previous class. So the overall objective of these classes is that not me saying everything, it's more of a discussion based session, so that most of us, including me, we can learn together. Okay, so it will be nice if you can just put your basic information, like what are you pursuing, from where you are, and all that stuff. Okay, now continuing our learning, as I've told you that yeah, I hope that you guys can see my presentation now and I am clearly audible. Yeah, so don't worry, those who have joined late, the video will be uploaded soon. And in the previous class I haven't discussed much, I have just discussed the course handout and some basics about cell membrane, so you have not lost much. And today we have just started with this video itself, so that you guys have a brief overall view of a cell membrane. Regarding the nitty-gritties of a cell membrane, we will see throughout the session. So one more particular video which I want you guys to see, apart from the one which we have just seen. I hope that things were pretty clear. As you know, membrane is a dynamic structure, right? So these were some of the drawbacks of the previous models. For example, Pauci molecular model, that they basically told that the membrane is static, but if the membrane is static, then it won't be able to do most of its function, right? You also know that throughout the temperature, the membrane's liquidity and the fluidity changes, right? So all this comes from the property that membrane is actually a dynamic structure and not a static structure. So the more you understand it as a dynamic process, it will be easier for you to like imagine the functions and the properties which are associated with the membrane in a better manner. Okay, so another video which I want you guys to see, again it will tell you about the membrane itself, but from a different perspective. Okay, so just let me share my screen. I hope that you guys can see my YouTube and
ink from Maybelline,
it's
New York.
pretty audible to
Instant
you
shine
guys.
color that lasts up to 16 hours.
All cells are completely surrounded by a membrane that separates them from their environment, kind of like how your skin completely covers your body. This cell membrane is sometimes called the plasma membrane, and it acts as a barrier, regulating what gets in and out of the cell. Because the membrane lets some things through but not others, we say that the membrane is semi-permeable or selectively permeable. If we zoom in on the membrane, we can see that it is made of several molecular parts. Most of the membrane is made of molecules called phospholipids. Phospholipids have a head made of phosphate and two tails made of fatty acids. The heads are hydrophilic, meaning that they are attracted to water, or literally water-loving. The tails, however, are hydrophobic, meaning that they repel water. Literally, hydrophobic means water-fearing. Because of these properties, phospholipids form two layers known as a phospholipid bilayer, with the hydrophobic tails facing each other and the hydrophilic heads facing the watery external environment and the watery cytoplasm inside of the cell. So the water lovers get to be next to water, and the water fearers can hide away from it. A perfect arrangement. The phospholipid bilayer is the main component of the membrane, and it is largely responsible for making the membrane semi-permeable. Basically, the smaller and less charged something is, the easier it is to pass through the phospholipid bilayer. Bigger, more charged things have a harder time. A second membrane component is cholesterol. Although people often think of cholesterol as a bad thing, it's actually a very important part of cell membranes, where it helps to stabilize the membrane and make it better at controlling what can pass through it. Proteins are another important part of the membrane. They do many functions, but one of the most important ones is transporting materials across the membrane, especially if they have a hard time passing through the phospholipid bilayer. A fourth membrane component is carbohydrates. These are often involved in cell identification. Blood types, for example, are the result of membrane carbohydrates attached to proteins. These help you to identify blood that belongs to you and distinguish it from foreign invaders. All of these components work together to make the plasma membrane that surrounds all cells, a critically important barrier needed for all life on Earth. If you enjoyed this video, please like it.
Yeah, so I guess now you guys have some basic idea about the membrane and the components of a membrane, right? So this is a very generic representative diagram of a cell membrane. So if you have heard the videos properly, you know what are the major components of a cell membrane. It's the lipid bilayer, indicating that the membrane is made up of lipid, right? And the lipids are of a particular type called phospholipids. So maybe in the next class, which will be on Saturday, we're gonna discuss about the different types of lipids which are present in a cell membrane. We're not gonna go too much detail into the structural property of a lipid, but we should know what are different types of lipids which are present in the membrane, because the different type of lipids which are present, they give the specific property of a membrane, whether the membrane will be more fluid or whether the membrane will be more rigid, or what kind of proteins will be associated with what kind of membrane, right? So all these things can be understood if only we know what are the lipids which are present in the living system, which make up the plasma membrane or the cell membrane. So if we talk about the generic structure of a cell membrane, we can clearly see that this is the outside of a cell, this is the inside of a cell, right? By outside and inside, why we are saying that this is outside and this is inside? Because the membrane is asymmetric in nature. What I mean by asymmetricity? Asymmetric means the upper leaflet, as I said, it's a lipid bilayer, right? So one bilayer means two layers, so each layer is called leaflet or leaf. So one leaf of a bilayer is different from the other leaf of a bilayer, hence the membrane is of asymmetric in nature. And the asymmetry comes from, so what asymmetry means? Asymmetry means that they both are not similar, though they are made up of lipids, they are made up of similar kind of lipids, yet they are different. So from where the asymmetry comes from? The asymmetry comes from the type of lipid and the concentration of lipid which is present in one leaflet, and it is different from the concentration of a lipid or a type of a lipid which is present in the other leaflet. In the coming classes, we'll see that there are certain lipids. After learning different types of lipid which are present in the bilayer, we'll see that there are certain type of lipids which have higher propensity of living in the lower leaflet, meaning the leaflet which is facing the cytosol of a cell, and there are other lipids which are commonly present in the other leaflet, that is the leaflet of a membrane which is facing towards the extracellular matrix. So the asymmetry in a membrane comes from the lipid, first thing. Second thing, the asymmetry in a membrane also comes from protein. As in the video lecture you have seen that the membrane has proteins and lipids and cholesterol also, right? So the proteins which are present in the upper leaflet and the proteins which are present in the lower leaflet, they also vary. Okay, hence the asymmetry in a biological cell membrane, which is lipidic bilayer in nature, comes from both the components, that is the lipid as well as the protein. Okay, so if we look at the basic structure of a cell membrane, we can clearly see that there are glycolipids. So what do we mean by glycolipids? Glycolipid means carbohydrate attached to the lipid. As you can see, this is the lipid head group, right? So these are phospholipids. The round structure is the head group, which is polar in nature, and this yellow part is called the tails, right? Phospholipid. So we're gonna discuss the structure of a phospholipid in the coming session. So if the carbohydrate is attached with the lipid head group, that is called glycolipid. Okay, if carbohydrate is attached with the protein which is present in the membrane, it is called oligosaccharide chain of glycoproteins. So glycolipid and glycoprotein, both glycolipid and glycoprotein, they are characteristic of a cell, hence where they should be predominantly present will be available towards the outside of a cell or towards the inside of a cell. They'll be present towards outside of a cell, right? Because they are providing the identity to a cell. If you remember, there are certain surface markers, there are receptors of a cell through which we determine what type of a cell we are dealing with, right? So those characteristics or specificity for a cell is determined by the carbohydrates which are associated with the cell. Those carbohydrates can either be associated with the lipid of a cell as well as with the proteins which are associated with the membrane. Okay, then GPI anchored proteins, we're gonna discuss all, we're gonna spend a lot of time in understanding the type of proteins and protein structure in general. So for now, just take it by its name. So GPI anchored proteins are the proteins which are associated with the plasma membrane or the membrane, and they have a linker molecule which links the protein, this purplish structure, to the membrane. Okay, this linker is called GPI. Then sterols, like cholesterols and they are present in the membrane, they provide the stability and rigidity to the cell membrane. As I have said that these blue ones are the phospholipid polar heads. There are different types of protein, and we're gonna discuss these type of protein when we talk about the fluid mosaic model, right, which is the most advanced and most accurate model predicting the structure of a membrane. So a protein which crosses both the leaflet or which crosses the lipid bilayer is called integral membrane protein, right, because they are part of the membrane, the interior of a membrane. There are certain proteins which are associated only with one side of a leaflet. They are called peripheral proteins. Okay, there are proteins which are directly associated only with one leaflet of a lipid bilayer, but they are associated with certain type of linkers. They are also peripheral proteins, but they are covalently linked with the lipid. So these are some major different types of protein, and this is just the schematic and very basic representation of a cell membrane. Okay, so talking about the functions, like if we are talking about the structure of a cell membrane, that's so dynamic, it's functionally relevant, it connects outside to the inside of a cell, but still what are the major functions associated with the cell membrane? So as we have discussed earlier, cell membranes are the barriers, right, and they're selectively permeable, so definitely they'll provide a barrier property to anything they are surrounding, and here we are calling the cell to which that boundary is present, cell membrane. Okay, so membranes are not merely a passive barrier, means they are not just lying there, right, they are functionally dynamic in nature and they are active. Passive means there is no association of energy and everything is flowing according to the concentration, but no, we know that it is not true, hence it is active in nature. They are flexible, self-repairing and selectively permeable. So they are flexible obviously because as we have seen, the membrane is made of lipid bilayer, and those lipid molecules, the phospholipids which we are talking about, phospholipids are connected to each other with multiple van der Waals interaction, hydrogen bonds, such things, right? So they are dynamic in nature, they are constantly breaking and constantly forming, hence it is flexible. Self-repairing, as in the video you have seen that there are certain molecules which are transported from one cell to the another cell, as like it can be anything, for example interleukins or chemokines, whatever is being secreted outside the cell, right? So it is exocytosis, endocytosis, so there is constant change in the concentration of a lipid of a membrane. So if something is pinched out of the cell membrane needs to replenish it, right? Hence that is why it is called self-repairing. And selectively permeable, as we have already discussed, small molecules, gaseous molecules, non-polar molecules, they can cross through the lipid membrane, but there are certain big molecules such as amino acids, ethanol molecule, polar compounds, which find it difficult to cross across the membrane. So if I pose this question that why polar molecules are facing issue in transversing through the membrane, so what do you guys think is the answer? Like feel free to put it in a chat box, otherwise obviously I'm gonna give the answer. It's a very simple basic chemistry phenomenon which plays a critical role in making the membrane selectively permeable. So I'll give you guys, if you are interested, you can also unmute yourself and give the answer, otherwise I'm gonna tell. So the question is why the membrane is selectively permeable and why it cannot allow all the molecules to pass through it. If for example carbon dioxide and oxygen can pass through the membrane without any issue, right, then why do we need the presence of proteins, as we have seen earlier, like today itself, that large molecules cannot pass through the membrane. I guess most of you might have some idea about it, but the so I'll help you out in getting to the answer. The answer is, if you remember in chemistry, there's a very basic phenomena called like dissolves like. Okay, so as you have seen, membrane is made up of what? Membrane is made up of lipids which have two distinct parts. First is the round, this blue round head groups which are polar in nature, and these yellow tails which are non-polar or hydrophobic in nature, right? So if a molecule is polar, imagine this, it will effectively interact with the head group. Like dissolves like. But the moment it starts entering through the membrane, the tails are hydrophobic in nature, right? So they won't allow polar molecule, because this is non-polar environment. So in non-polar environment, how come a dislike property molecule which is polar in nature, opposite to the basic characteristic of the interior of a membrane, will be able to pass? So it will face a lot of energy barrier, lot of huge energy for a polar molecule to pass through the lipid bilayer. Okay. Similarly, if the molecule is non-polar in nature or hydrophobic in nature, right? So it will initially face high energy barrier here itself. Why? Because heads are polar in nature, hence they will reflect and not attract. Okay. That is why the membrane is selectively permeable. It will allow small molecules to pass through them easily, because the energy associated with small non-polar molecules is very less, and it can be provided by the system itself. On the other hand, if the molecule is big and has a polar property attached to it, it cannot directly pass through the membrane and needs specialized proteins for such functionality, which leads to the selective permeable nature of a membrane. Okay, so their flexibility permits the shape changes that accompany cell growth movement, such as amoeboid movement. With their ability to break, two membranes can fuse, as in exocytosis, or a single membrane enclosed compartment can undergo fission to yield two sealed compartments, as in endocytosis or cell division, without creating gross leaks through cellular surfaces. I hope this is self-explanatory point. Then their selective permeability allows them to serve as molecular gatekeepers, right? And it's a very important property, because through this property only the cell assures that its survival, whether it needs something from outside, whether it needs something to be ejected outside the cell, everything is regulated by this property of a membrane, which is selectively permeable. These membrane functions are accomplished primarily by a broad array of proteins and enzymes in and on membrane. We're gonna discuss about these, some like different types of protein and its functionality. At the cell surface, transporters move specific organic solutes and inorganic ions across the membrane. Receptor senses extracellular signals and trigger molecular changes in cell. Ion channels mediate electrical signaling between cells, and adhesion molecules hold neighboring cells together. Again, this is just the overview of the functionality of a membrane. So before going more deep into the functionality of a cell, as we have seen a very generic structure of a cell membrane, after that we're gonna discuss about the fluid mosaic model also. But before that, I would like you guys to see another video which will describe the basic structure of a plasma membrane, and then we can discuss about the fluid mosaic model in the next session. Okay, I hope you guys can see my YouTube and can hear.
I'm going to talk about the plasma membrane. The plasma membrane of cells is made up of something called phospholipids. And here is a typical structure of a phospholipid. The phospholipid is made up of three subunits. It has a phosphate group, a glycerol molecule, and two fatty acid tails. The head of the phospholipid is the phosphate group, and it's polar, which means that it's hydrophilic. Hydro means water, phil means love, so it loves water. The tails are nonpolar, and so they are hydrophobic. They don't like water. they tend to stay away from water. So what we have here is known as the phospholipid bilayer, which makes up the cellular membrane. On the inside of the cell is an aqueous environment, an environment that contains water. And the same is true on the outside of the cell. On the outside of the cell, rather. Notice that the heads of the phospholipids are oriented in such a way that they're facing water. It's because that they're polar. They're attracted to it. Whereas the interior are the tails of the phospholipids, and they're directed away from water because that region is hydrophobic. It's nonpolar. So that's the structure of the phospholipid bilayer. It's made up of phospholipids, and that comprises the cellular membrane or the plasma membrane. Now let's talk about the structure of the phospholipid. So the phospholipid, as we said before, it's made up of a glycerol molecule. And it contains two fatty acid nonpolar tails. In this example, the first fatty acid, which is this one here, is a saturated fatty acid because it doesn't have any double bonds. The second one is an unsaturated fatty acid because it has a double bond. Now the third part is the phosphate group. The phosphate group is attached to two carbons, which is also attached to a nitrogen atom with three other methyl groups on it. The nitrogen atom has a positive formal charge, and this oxygen has a negative formal charge. So that's the typical structure of a phospholipid. So this part here is the head of the phospholipid. That is the polar part. As you can see by the formal charges, it's going to be attracted to water. So it has a phosphate group, two carbons, and a trimethyl amine group. Well, this is technically an ammonium group since there's 4 carbons attached to that nitrogen atom. And these two fatty acid chains that are part of the phospholipid, that is the hydrophobic part of the molecule. So those are the two tails. That's the part that is oriented away from water. And in the middle, we have a glycerol molecule. I'm not sure what just happened there.
Yeah, so as you have seen that these are some was a very basic structure of a phospholipid, and as I have mentioned, I'm in the coming class I'm gonna discuss more about the different types of lipid, and maybe we can discuss some basic structure of a phospholipid, because though there are different types of phospholipids, the basic structure or the backbone of a phospholipid will remain the same, which we have just seen, right? That there is a polar head and then there are tails which are associated with it. In the polar head itself, if you change the functional group, we can change it to different lipids, and hence different lipids, different functional group will provide different property to the membrane. Okay, so for today I guess we gonna wrap it up here. On Saturday I'm gonna circulate the link as usual, and the class will be from 3 to 4 most probably, otherwise I'll intimate you in advance. So if you guys have any question, feel free to ask or type them in the chat box. I can see, okay, hi Shrish, you are pursuing BSc honors in biotechnology, you are in 3rd year. Okay, great. So hope if you guys have any question, shoot, and if not, then that is all from my side, especially with respect to today's session. So if there are no questions, is there any teams group we can create if you want? I haven't generated any such link, though I have provided you guys my email address. Feel free to drop your queries or ask for anything there itself. I'm pretty active on my mail ID. Cool. So I'll take your leave now. Have a good day and thank you for joining in. Bye bye. No problem.